So in this discussion, we are going to talk about another type of passive transport, which is osmosis. So the question we're really asking here is, how does water move across the cell membrane? So osmosis is specific to water, so that's what we really need to keep in mind here. So osmosis is the movement of water molecules across the cell membrane. So you can see here we've got two types of molecules. We've got all these blue dots here. These are the water molecules. Okay, these are the water molecules. And then we've got the red dots here. These ones here are the solute molecules. Maybe we should write that in. So these ones here, these are solute molecules, whereas these ones here, these are H2O. And here we've got some sort of selective membrane, and it only lets water molecules through. So this membrane here is only letting, this one here is only letting water molecules pass through. Right? Only water molecules are getting through this membrane here. And you can see that there's these little spaces here. Those are the pores that are letting the water molecules through. But the solute molecules, these solute molecules are not able to pass through. So what you need to remember is that water molecules, they move from a low solute concentration to a high solute concentration. And the key word here is solute. Okay, you have to describe which side has the high solute concentration and which side has the low solute concentration. And water will always move from low solute concentration to high solute concentration. And in osmosis, what you need to remember is that, or in an ideal situation, when we're describing osmosis, the solute molecules are not crossing. In reality, some solute molecules are able to pass across a membrane. Both water and solute molecules are both moving across the membrane. But in this kind of controlled example of like perfect osmosis, this membrane here, this semi-permeable membrane right here, this selectively permeable membrane, is not permeable to the solute. It's only soluble or only permeable to water. Okay, it's only permeable to water. Oh, we don't want that color. Only permeable to water. So we should probably discuss what the word permeable means. Permeable is just another way of saying to let something go through. So here you can see the water molecules are moving it through that membrane. Okay, so if we go back to this, picture here, you can see that on this side over here, there's 1, 2, 3, 4 solute molecules, and this volume this volume is relatively, is the same as the volume on the other side, right? So this volume here on this side is equal to the volume on this side over here. And so on the other side, we've got 1, 2, 3, 4, 5, 6, 7, 8, 9. We've got 9, so just a little bit more than 2 times the number of solute molecules on the right-hand side compared to the solute molecules on the left-hand side. And those two volumes are the same volumes. So that means that on the right-hand side, this would be the high solute concentration, or CONC, oh dear, high solute concentration. And on the left-hand side, this is the low solute concentration, CONC concentration. So this is the low solute concentration, this is the high solute concentration. Low solute concentration, high solute concentration. So what does that mean for water? Water molecules are going to move from the from the low solute concentration to the high solute concentration. They're going to move this way. Water molecules are mostly going to be moving in this direction. And they will go in that direction, and they're going to change the volume of water. So you can see here, we've got the solute molecules. So again, over here, if you look here, this was not very intelligent. They have made the solute molecules blue. So let's just go ahead and change that a little bit. So these solute molecules, let's make them red, okay? So you can see over on this side, there's lots of the solute molecules. And then on the other side, there's not as many solute molecules, right? There's only a few solute molecules on this side. And the water molecules, we don't see those. They're not really visible on here. So let's maybe make some water molecules. Let's use a different color. So the water molecules are between all those solute molecules here. Okay, and there's water molecules over here as well. But we don't really, it doesn't really matter how many water molecules there are necessarily, because we're more concerned about the total volume. And so what's going to happen is that over on this side here, this is the low solute concentration because it doesn't have as many red solute molecules. And this is the high solute concentration. Okay, so let's write this in here. This over here is the low solute concentration, and this is the high solute concentration. And so the water molecules are going to end up moving from low solute to high solute concentration. So they're going to move across the membrane this way. Water molecules are going to move across the membrane this way. And really what this is doing is it's making this side the same concentration as this side, and it does that by increasing the volume on this side. So if you look over here, now the volume on this side here, right, the volume over here volume A is greater than over here because this is occupying less space on the right-hand side. So the volume on the left-hand side is occupying more space. Volume B. So volume A now is greater than volume B, which means that this is going to cause the concentrations to become equal. Okay, so now this side, concentration A is going to be equal to concentration B. These are going to be equal. Okay, now if we look over here, here's side A, right? The orange side we're making it. Okay, so in this case here, at the beginning, volume A was equal to volume B. Look, those are the same, right? Those are the same volume. Volume A equaled volume B. But concentration, right? Concentration A was greater than concentration B. The total number of solute molecules hasn't changed, has it? You can see here that the number of solute molecules is the same still. It's just that the volume, they now occupy a greater volume. And so that has decreased the concentration, right? The number of solute molecules on the right over here has not changed, but the volume has decreased. And so they are now occupying less volume. So their concentration has changed. Remember that concentration is equal to the mass divided by the volume, right? So if we increase the volume, then we decrease the concentration, right? Let's write that in. Okay, so the mass, or the amount of solute has not changed, right? We didn't change this. The only thing that's changing, because water molecules are moving, is that we're changing the volume. And so remember, if we increase the volume, then you decrease the concentration. And opposite to that, if you decrease the volume, then you increase the concentration. Okay? So that's what's happened over here. Water molecules have moved into this higher concentration. They've moved from low solute concentration to high solute concentration. That caused the volume of this solution to increase. Therefore, the concentration decreased. Over here, the volume decreased, and so that increased the concentration. And this will happen. This side will increase in volume, and this side will decrease in volume until the concentrations are equal and they have reached dynamic equilibrium. Okay. So now we also need to be able to describe solutions. So when you're explaining osmosis, you need to we are always comparing two solutions. Okay, we're usually comparing the inside of the cell to the outside of the cell, or the cytoplasm relative to the extracellular environment, or you're comparing two sides, right? A to B on two sides of a membrane. So when you're comparing two solutions, the hypotonic solution has the lower solute concentration. Okay, hypo means less than. Hypo means less than, or lower. Okay, hyper means the opposite. It's more than. or greater. Okay? So in that case, with that being said, a hypertonic solution has the greater solute concentration. And if two solutions have the same concentration on either side of the membrane, then we call it being isotonic. Okay, when two solutions are isotonic, they have equal solute concentrations, and so there's no net movement of water. And the key word here is no net movement of water. What this means is that they reach a dynamic equilibrium, right? This is dynamic equilibrium. Okay, remember that water molecules are still moving in both directions, just equally in opposite directions, right? All right, now let's compare some solutions and then we'll get back to plant and animal cells. So these are two examples that you need to know. You need to be able to describe how osmosis functions in plant cells, and you also need to describe how it functions in animal cells. This is essentially an animal cell over here. It's a red blood cell. So this is animal cell examples, or the example. Okay, so you also need to be able to describe how this works in animal cells. Okay, so plant cells and animal cells. And we'll talk about that in a little bit. They behave a little bit differently. But first, let's make sure that we have a look here and learn a little bit about hyper, hypo, and isotonic. So if you imagine that each of these triangles represents 1 gram, okay, let's imagine that each triangle equals 1 gram of solute. Okay, and let's imagine that this volume, the volume is equal to 1 liter. Okay, 1 liter of solute. So in this case here, you've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. You've got 12 grams of solute per 1 liter of solution. So this would equal a 12 gram per liter solution. Okay, so that's the concentration, right? That's the concentration, right? Because it's mass divided by volume. Okay, so now what you need to do is you need to have a look at B, C, D, E, and F, calculate their concentrations, right? What are their concentrations? Concentration is equal to, concentration equal to, concentration equal to, right? And then once you've done that, you can compare them. So for example, let's compare solution A to solution C. So we already know that solution A has a mass, or has a concentration of 12 grams per liter. Let's have a look at solution C. 1, 2, 3, 4, 5. This one here, its concentration is equal to 5 grams per 1 liter. It equals 5 grams per liter, right? Simple. And so this is 5 grams per liter. So 12 is greater than 5. So this is a hypertonic. This solution is hypertonic to solution C, right? Solution A is hypertonic to solution C. So then you can practice 2, 3, and 4, okay? Make sure that you're using the words hypotonic, hypertonic, or isotonic. Okay, you need to use those words. Use those words. Hypertonic, hypotonic, isotonic. Which one fits on the line? Only one will apply. Now, if we're changing the volume, okay, you could do the same thing, right? So let's imagine, so now we're being explicit about the volume, right? So we're changing the volumes here. So now this one here is solution A, I think it's the same, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Oh, no, it's not. So this one here is 10. The concentration is equal to 10 grams per 1 liter, which equals 10 grams per liter. This one over here, though, 1, 2, 3, 4, 5. This is only 5 grams, right? The concentration is equal to 5 grams divided by, oh, look, the volume is half a liter, so 0.5 liters, which equals, oh, wait a minute, that's equal to 10 grams per liter. Oh, so solution A is isotonic to solution B. Okay, it's isotonic because they have the same concentrations, 10 grams per liter, 10 grams per liter, right? So if they're equal, they're isotonic. So you should be able to compare, and I know that the questions aren't here, but you should be able to compare A to C, A to D, B to C, B to D, oops, and C to D. Okay, so you should be able to compare all of those and use the words, again, use the words isotonic, hypotonic, hypertonic to describe that solution, right? Solution 1 is iso, hyper, hypo to solution 2, right? So compare one solution to a second solution. All right. So we talked about how plant and animal cells have different responses to their environments. And so let's have a look at that. So we're looking at freshwater versus isotonic environments versus saltwater or high solute. So in other words, this, if we were to write out those words, oh yeah, we've got them here. We've got, this is a hypotonic solution. This is a hypertonic solution, salt water. And then isotonic is balanced, right? It's equal. Let's have a look at the freshwater first. So in this case here, if it's a hypotonic solution, an animal cell will swell and burst. So that's called lysing. So it will swell and burst. So animal cells, so this is animal cell. It will swell and burst. Okay, in other words, it will lysed. Okay, it means to be cut open. That's what that means. Now, in freshwater, in a hypotonic environment, plant cells, here's the plant cell down here, here's the plant cell. It will swell, but it will not burst. Okay, so plant cells prefer this because what ends up happening is that the water is pushing outwards. So the water will actually go into this part here, which is called the central vacuole, right? We learned that. This here is called the central vacuole. And so water will fill up into here, right? Water will actually move into the central vacuole, and the central vacuole will cause the cell to push out against the cell wall. Remember, this here is the cell wall. And the cell wall provides support. Okay, and the animal cells don't have that, right? Animal cells, they don't have a cell wall. No cell wall. So that's why they burst instead of just swelling up and being kind of pressurized. They increase, they have this like nice internal pressure, right? So that pressure, okay, pressure builds up. And that pressure is called being turgid, okay, or turgor. It creates turgor pressure. And plants prefer that. This is their preferred state. Okay, in an isotonic environment, water is moving in and out of the cell equally, okay? And this is the preferred state for animal cells. So animals prefer this. This is the preferred, let's call it the preferred condition for animal cells. Plant cells don't like this though, because the central vacuole, right, loses pressure. So the turgor pressure decreases, right? It decreases. And for that reason, it becomes flaccid or floppy. It becomes floppy. Now, both plant and animal cells do not like a hypertonic environment. They don't like a hypertonic environment because what happens is that water leaves the cell. And so the cell shrinks, right? So the cell shrinks here and can die. Okay, so that's not optimal for cells. They don't want to lose too much water. Plant cells, though, they shrink. They shrink, and the cell membrane pulls apart from the cell wall. Okay, and that's called being plasmolyzed. That's called being plasmolyzed. Okay, or the process is called plasmolysis. Okay, plasmolysis. Plasma means like the liquid inside the cell. So the why am I drawing a blank on this? The cytoplasm, right? The cytoplasm is pulling away, or is the cytoplasm, the plasma, the liquid inside the cell is being cut from the cell. So it looks like it's breaking up. And so that's because the cell membrane is pulling away from the cell wall. Okay, so there's a few ways that cells can kind of get around this. In fresh water, living things like a paramecium. So here's a paramecium. Remember, in fresh water, they're going to swell up and burst. This is a protist. This is an animal-like protist. Okay, so it's kind of like an animal. And so it prefers isotonic environments, but they live in fresh water. So the problem is that water is going to move into the cell. And so the paramecium is going to swell and burst if it doesn't get rid of its water. But that's what it does. It has this organelle right here called a contractile vacuole. It's a specialized vacuole. And its job is to squeeze and shoot the water out of the cell. So it's going to take water and blast it out of the cell. So it's going to remove water from the cell. And so the cell doesn't swell up and explode. Okay, but remember, plant cells are happy in this environment. Plant cells are happy. This is just the paramecium here, You can see the contractile vacuole is full of water here, and here it's squeezed all the water out, and so it's empty now. In salt water, remember that plant and animal cells don't appreciate being in salt water because they shrink and die. Or a plant cell will undergo plasmolysis, right? So the plant cell doesn't want to be plasmolized, and the animal cell doesn't want to shrink and shrivel up and die like a raisin. So the cell is losing too much water is the problem, right? And some animals that this happens in is shellfish, so like clams and oysters and whatnot. There are some like mangrove trees live in semi-salty water, kind of like fresh water and salt water mixed, but it's still too salty. And so the problem is that these cells are, these plant cells and animal cells are losing too much water, right? And so in plant cells, you'll get plasmolysis. The cell membrane here separates from the cell wall, which is right here, right? And in animal cells, they shrink and die. So the shrinking cell is not good. So the solution is that they just have proteins in their membranes that take water into the cell, right? Bring water, what this means is they bring water into the cell. Okay, they use active transport to do that, which we'll talk about later. And then just an example of how balanced solutions are really important is when you go to the hospital. So here we've got someone who's in the hospital, and you can see that they've got this needle that's going into their blood here, right, into their veins. And so what we have to be mindful of is that this solution, right, this solution here is called standard saline solution. And it must be isotonic to our red blood cells. Because if it's hypotonic, then our red blood cells will swell up and burst, right? Our red blood cells will swell and burst. And if it's hypertonic, then our red blood cells will shrink and die. And so that's a big problem. So we need to make sure that the solution here is isotonic so that our red blood cells keep their shape. Okay, that's really important. Plant cells don't like this. They lose their turgor pressure, and so they become flaccid, but they are okay. They can still survive in that environment. If we go back to the red blood cell example back here, you can see here's what happens to red blood cells. Here's a happy red blood cell. It looks like a ring or a donut. Here's an unhappy red blood cell in a hypertonic solution. You can see it's shriveling, it's shrinking up, and it's going to dry up and turn into a raisin and die. That's not good. And you can see here the red blood cell is swelling up. It's increasing its volume, and it's lost this little dimple here. And so it doesn't have that center kind of indent. And this is also not good. It will explode and die. So that happens if we have a hypotonic solution going into our blood. So that is osmosis and some examples that are related to osmosis.